Structure of atoms, molecules. 1. Elements consist of only one kind of atom and cannot be decomposed into simpler substances. Our planet is made up of some 90 elements. Tiny amounts, sometimes only a few atoms, of additional elements have been made in nuclear physics laboratories, but they play no role in our story. Of these 90, only 25 or so are used to build living things. 2. The table shows the 11 most prevalent elements in the lithosphere, the Earth's crust, and in the human body. 3. Living is only a fraction of the elements available to it, but, as the table shows, the relative proportions of those it does acquire from its surroundings are quite different from the proportions in the environment. So, the composition of living things is not simply a reflection of the elements available to them. For example, hydrogen, carbon, and nitrogen together represent less than 1% of the atoms found in the Earth's crust but some 74% of the atoms in living matter. One of the properties of life is to take up certain elements that are scarce in the non-living world and concentrate them within living cells. Some sea animals accumulate elements like vanadium and iodine within their cells to concentrations 1,000 or more times as great as in the surrounding seawater. It has even been proposed that uranium be mined from the sea by extracting it from certain algae that can take up uranium from seawater and concentrate it within their cells. 4. There is still some uncertainty about the exact number of elements required by living things. E.g., selenium and aluminum, are found in tiny amounts within living things, but whether they are playing an essential role, selenium is, or simply an accidental acquisition, aluminum probably, is sometimes difficult to determine. 5. Atoms. Each element is made up of one kind of atom. We can define an atom as the smallest part of an element that can enter into combination with other elements. Each atom consists of a small, dense, positively charged nucleus surrounded by much lighter, negatively charged electrons. 6. The nucleus of the simplest atom, the hydrogen atom H, consists of a single positively charged proton. Because of its single proton, the atom of hydrogen is assigned an atomic number of 1 and a single electron. 7. The charge of the electron is the same magnitude as that of the proton, so the atom as a whole is electrically neutral. Its proton accounts for almost all the weight of the atom. 9. The nucleus of the atom of the element helium, He, has 2 protons, hence helium has an atomic number of 2 and 2 neutrons. Neutrons have the same weight as protons but no electrical charge. The helium atom has 2 electrons so that, once again, the atom as a whole is neutral. 10. The structure of each of the other kinds of atoms follows the same plan. From lithium at no equals 3 to uranium at no equals 2, the atoms of each element can be listed in order of increasing atomic number. There are no gaps in the list. Each element has a unique atomic number and its atoms have 1 more proton and 1 more electron than the atoms of the element that precedes it in the list. Electrons. 1. Electrons are confined to relatively discrete regions around the nucleus. The 2 electrons of helium, for example, are confined to a spherical zone surrounding the nucleus called the K shell or K energy level. 2. Lithium at no equals 3 has 3 electrons, 2 in the K shell and 1 located farther from the nucleus in the L shell. Being farther away from the opposite plus charges of the nucleus, this third electron is held less tightly. 3. Each of the following elements, in order of increasing atomic number, adds 1 more electron to the L shell until we reach neon at no equals 10, which has 8 electrons in the L shell. 4. Sodium places its 11th electron in a still higher energy level, the M shell. 5. From argon, this shell is gradually filled with electrons until, once again, a maximum of 8 is reached. Note that after the K shell with its maximum of 2 electrons, the maximum number of electrons in any other outermost shell is 8. As we all see, the chemical properties of each element are strongly influenced by the number of electrons in its outermost energy level shell. 6. This table shows the electronic structure of the atoms of elements 1 to 36 with those that have been demonstrated to be used by living things. 4 elements of still higher atomic numbers that have been shown to be used by living things are also included. 7. The electronic structure of an atom plays a major role in its chemistry. The pattern of an atom, especially those in the outermost shell, determines the valence of the atom, that is, the ratios in which it interacts with other atoms, and to a large degree, the electronegativity of the atom, that is, the strength with which it attracts other electrons. 8. Elements with the same number of electrons in their outermost shell show similar chemical properties. Elemental composition of the lithosphere in the human body. Each number represents the percent of the total number of atoms present. For example, 47 of every 100 atoms found in a representative sample of the lithosphere are oxygen while there are only 19 atoms of carbon in every 10,000 atoms of lithosphere. Composition of the lithosphere. Composition of the human body. Oxygen 47, hydrogen C3, silicon 28, aluminum 7.9, carbon 9.5, iron 4.5, nitrogen 1.4, calcium 3.5, calcium 0.31, sodium 2.5, phosphorus 0.22, potassium 2.5, chlorine 0.03, magnesium 2.2, potassium 0.06, titanium 0.46, sulfur 0.05, hydrogen 0.22, sodium 0.03, carbon 0.19, magnesium 0.01, all others less than 0.1, all others less than 0.01. Example 1. Fluorine, chlorine, bromine, and iodine each have 7 electrons in their outermost shell. These so-called halogens are also quite similar in their chemical behavior. When dissolved in water, for example, they all produce germicidal solutions. Example 2. Those elements with 1, 2, or 3 electrons in their outermost shell are the metals. Example 3. Those elements with 4, 5, 6, or 7 in their outermost shell are the nonmetals. Example 4. Helium with its 2, neon, argon, and krypton each with 8, have filled their outermost shells. They are the so-called inert or noble gases. They have no chemistry at all. Under normal conditions they do not interact with other atoms. So, it is the number and arrangement of the electrons in the atoms of an element that establish the chemical behavior of that element. 9. The atoms of an element interact with other atoms in such ways and ratios that they can fill their outermost shell with 8 electrons, 2 for hydrogen. They may do this by acquiring more electrons from another atom, losing electrons to another atom, sharing electrons with another atom. The number of electrons that an atom must acquire, or lose, or share to reach a stable configuration of 2 for hydrogen is called its valence. 10. Hydrogen, lithium, sodium, and potassium atoms all have a single electron in their outermost shell. Fluorine, chlorine, bromine, and iodine atoms all have 7. Any atom of the first group will interact with a single atom of any of the second group forming HCl, NaCl, Ke, etc. The result of these interactions is a pair of atoms each with an outermost shell like that of one of the inert gases, 2 for hydrogen, 8 for the others. Atoms with 2 electrons in their outermost shell interact with chlorine and the other halogens to form, e.g., BCl2, MgCl2, CaCl2. Again, the result is a pair of atoms each with a stable octet of electrons in its outermost shell. The element with 3 electrons in their outermost shell will interact with chlorine in a ratio of 1:3, forming BCl3, AlCl3. Carbon atoms, with their 4 electrons in the L shell, interact with chlorine to form CCl4. Nitrogen, with its 5 outermost electrons, interacts with hydrogen atoms in a ratio of 1:3, forming ammonia, NH3. Oxygen and sulfur, with their 6 outermost electrons react with hydrogen to form water, H2O, and hydrogen sulfide, H2S. 11. What determines whether a pair of atoms swaps or share electrons? The answer is their relative electronegativities. 2 atoms differ greatly in their affinity for electrons, that is, in their electronegativity, then the strongly electronegative atom will take the electron away from the weakly electronegative one. Example 1. Not weakly electronegative gives up its single electron to an atom of chlorine, strongly electronegative, to form NaCl. The sodium atom now has only 10 electrons but still 11 protons, so there is a net positive charge of 1 on the atom. Similarly, chlorine now has 1 more electron than proton, so it now has a net negative charge of 1. Electrically charged atoms are called ions. The mutual attraction of opposite electrical charges holds the ions together by ionic bonds. Example 2. Carbon and hydrogen are both only weakly electronegative, so neither can remove electrons from the other. Instead, they achieve a stable configuration by sharing their outermost electrons, forming covalent bonds of CH4. Isotopes. 1. The number of protons in the nucleus of its atoms, which is its atomic number, defines each element. However, the nuclei of a given element may have varying numbers of neutrons. Because neutrons have weight about the same as that of protons, such atoms differ in the atomic weight. Atoms of the same element that differ in their atomic weight are called isotopes. Atomic number element. Energy levels or Ne2816229 copper Cu. Ne28162. This atom is designated carbon-12 or 12C. It is arbitrarily assigned an atomic weight of 12 daltons, named after John Dalton, the pioneer in the study of atomic weights. Thus a dalton is 1/12 the weight of an atom of 12C. Both protons and neutrons have weights very close to 1 dalton each. Carbon-12 is the commonest isotope of carbon. Carbon-13C with 6 protons and 7 neutrons, and carbon-14, 14C with 6 protons and 8 neutrons are found in much smaller quantities. 3. Isotopes, tracers, one can prepare, for example, a carbon compound used by living things that has many of its normal 12C atoms replaced by 14C atoms. Carbon-14 happens to be radioactive. By tracing the fate of radioactivity within the organism, one can learn the normal pathway of this carbon compound in that organism. Thus 14C serves as an isotopic label, or tracer. 4. The basis of this technique is that the weight of the nucleus of an atom has little or no effect on the chemical properties of that atom. The chemistry of an element and the atoms of which it is made, whatever their atomic weight, is a function of the atomic number of that element. As long as the atom had 6 protons, it is an atom of carbon irrespective of the number of neutrons. Thus while 6 protons and 8 neutrons produce an isotope of carbon, 14C, 7 protons and 7 neutrons produce a totally different element, nitrogen-14. A2. Chemical bonds. All interactions between atoms are electrical attractions between charges. Ionic and covalent bonds hold atoms together to form molecules. Weak bonds, hydrogen, van der Waals and other types, hold molecules together. 1. Ionic bonds. Compounds with ionic bonds split into ions in water. Ions conduct electricity, give specialized cells, nerve, muscle, give specialized cells, nerve, muscle, excitable properties. Suppose Na gives one of its electrons to Cl, the Na now has a plus charge and the Cl will have a charge. These charged atoms are referred to as ions, and since they have opposite charges they attract each other and form a chemical bond, they form NaCl, common table salt. Positively charged ions equals cations, i.e., Na plus. Negatively charged ions equals anions, i.e., Cl. 2. Covalent bonds. Each atom donates one or more electrons to the bond. Bond electrons spend most of their time between the two atoms, attracting both nuclei and pulling them together. If each atom donates 2 electrons to a bond, a double bond is formed. Double bonds are stronger and more rigid than single bonds. A triple bond is formed when each atom donates 3 electrons to the bond. This type of bond holds together the long chains of macromolecules. These molecules do not split apart in water. 3. Hydrogen bonds occur when a hydrogen ion is sandwiched between 2 atoms, usually nitrogen and oxygen. It is weaker, about 25 times, than covalent or ionic bonds and mainly occur between molecular groups with permanent dipoles. In water, hydrogen bonds makes water molecules stick together. It is responsible for many of the strange properties of water such as cohesion, high boiling point. Hydrogen bonds cause protein chains to spiral and bend, giving unique shapes. In DNA they hold together the 2 chains to form the double helix. Allow chains to unzip for replication and transcription. 4. Van der Waals and other weak bonds. Weak forces that can bond like atoms together. They are especially important between chains of carbon atoms. They are, although weak, numerous bonds between the chains can add up to produce significant cohesion. Such bonds determine physical state of compounds, gas, liquid or solid, and occur when one atom induces a temporary dipole in another atom. These are important in holding like molecules together. Often determine the solid, liquid or gas state of a compound. Saturated fats are solid at room temperature because they have more van der Waals attractions than unsaturated fats, which are liquid. B1. Composition, structure and function of carbohydrates. Carbohydrates are mainly compounds of carbon, hydrogen and oxygen. Carbohydrates are so called because in most of them, the proportion of hydrogen and oxygen is the same as in water, H2O, i.e., 2, 1. Their general formula is CxH2xOX. These are also known as saccharides, compounds containing sugar. Carbohydrates are produced by green plants during photosynthesis. These constitute about 80% of the dry weight of plants. Carbohydrates are divided into 3 main classes: monosaccharides, derived monosaccharides and oligosaccharides. Monosaccharides. These have single saccharide units which cannot be hydrolyzed further into still smaller carbohydrates, have general formula CnH2ON. These are 3 to 7 carbon atoms, and are classified according to the number of C atoms as trioses, 3C tetroses, 4C pentoses, 5C hexoses, 6C and heptoses, 7C. Of these, pentoses and hexoses are most common. Monosaccharides are important as energy source and as building blocks for the synthesis of large molecules. All monosaccharides are either polyhydroxyaldoses or ketoses. The 2 simplest monosaccharides are trioses, e.g., glyceraldehydes and dihydroxyacetone. Tetroses, e.g., erythrose, are quite rare. Erythrose takes part in the synthesis of lignin and anthocyanin pigments. Ribulose, xylulose and arabinoses are pentoses. Xyluloses and arabinoses polymerize to form xylins and arabins, which are cell wall materials. Glucose, fructose, mannose, galactose are hexoses. These are white, sweet-tasting, crystalline and extremely soluble in water. Glucose is a universal sugar. It is also known as dextrose or grape sugar or corn sugar. Fructose is the most common form of sugar fruit. It is also known as levulose. It is the sweetest among naturally occurring sugars. Monosaccharides have free aldehyde or ketone group which can reduce Cu++ to Cu+. Hence, these are also called reducing sugars. 2. Derived monosaccharides. Deoxy sugar, loss of oxygen atom from ribose yields deoxyribose, a constituent of DNA. Amino sugar, monosaccharides having an amino group, e.g., glucosamine, galactosamine. Sugar acid, e.g., ascorbic acid, glucuronic acid, galacturonic acid. Sugar alcohol, e.g., glycerol and mannitol, present in brown algae. 3. Oligosaccharides. The sugars with limited numbers, 2 to 10, of monosaccharides are called oligosaccharides. These include trisaccharides, tetrasaccharides, hexasaccharides, heptasaccharides, etc. Disaccharides. These are formed by condensation reactions between 2 monosaccharides, usually hexoses. The bond formed between 2 monosaccharides is called a glycosidic bond. It normally forms between C atoms 1 and 4 of neighboring units, 1, 4 bond. Once linked, the monosaccharide units are called residues. Maltose, glucose plus glucose, lactose, glucose plus galactose, sucrose, glucose plus fructose are most common disaccharides. Sucrose is most abundant in plants and is known as cane sugar or table sugar. It is the sugar we buy from market. B. On hydrolysis, disaccharides release their respective constituent monosaccharides, e.g., hydrolysis of sucrose yields 1 molecule each of glucose and fructose. Disaccharides. Sugars composed of 3 monosaccharide units are called trisaccharides, e.g., raffinose. Raffinose is a common saccharide found in plants. Upon hydrolysis, it yields 1 molecule each of glucose, fructose and galactose. Larger oligosaccharides are attached to the cell membrane and cell recognition is due to their presence. They also take part in antigen specificity. Polymerization of a large number of small molecules results in the formation of large molecules of high molecular weights, which may be branched or unbranched. These are called macromolecules. These include some polysaccharides, proteins and nucleic acids. 4. Polysaccharides. These are polymers of monosaccharides and are branched or unbranched linear molecular chains. These are insoluble carbohydrates and are considered to be non-sugars. Starch, glycogen, cellulose, pectin, hemicellulose, inulin and polysaccharides. 5. Polysaccharides are of 2 types. Homopolysaccharides consists of only one type of monosaccharide monomer, e.g. starch, glycogen and cellulose, fructin, xylin, arabin, galactin. Heteropolysaccharide consists of more than one type of monosaccharide monomer, e.g. chitin, agar, arabinogalactans, arabinoxylin, etc. 6. Polysaccharide types, storage e.g. starch and glycogen, structural e.g. chitin, cellulose and mucopolysaccharides e.g. keratin sulfate, chondroitin sulfate, hyaluronic acid, agar, allergenic acid, carrageenan and heparin. 7. Storage polysaccharide. Starch, glycogen and inulin are reserve food materials. Starch turns blue with iodine. Starch is a polymer of glucose. It is the major reserve food in plants. Starch has two components, amylase, an unbranched polymer, and amylopectin, a branched polymer. Amylopectin consists of 2,200,000 glucose molecules forming straight chain and branches after 25 glucose units. Branching point has alpha 1 to 6 glycosidic linkage. Amylose consists of alpha 1 to 4 glycosidic linkage between alpha D glucose molecules. It is straight chain of 200, 1,000 glucose units. It is helical, each turn consists of 6 glucose units. Starch molecules accumulate in the form of layers, stratifications, around a shifting organic center, hilum, to form starch grains. In eccentric starch grains, hilum lies on one side. These are found in potatoes. In concentric starch grains, hilum is present in the. These are found in wheat, maize, and pea. These are found in wheat, maize, and pea. Dumbbell-shaped starch grains are found in the latex of euphorbia. Grains with single hilum are called simple, e.g. maize, but those with more than one hilum are called compound, e.g. potato, rice. Eat glycogen. Glycogen is the animal equivalent of starch. Many fungi also store it. Glycogen turns red-violet with iodine. It consists of 30,000 glucose units joined by alpha 1 to 4 bonds, much more branch than starch. Branching point has alpha 1 to 6 linkage. Branching occurs after 10 to 14 glucose units. Eat inulin. It is an unusual polysaccharide and polymer of fructose. It is used as a fructose, particularly in roots and tubers of the family Compositae, e.g. Dahlia tubers. 8. Structural polysaccharide. Cellulose. Cellulose is the main structural polysaccharide of plants. It is the most abundant molecule on Earth. It is almost confined to plants, though it is found in primitive fungi and lower invertebrates also. It is structural component of all plant cell walls, constituting, on an average, about 20–40% of the wall. Cotton fibers contain the largest proportion, 90%, of cellulose among natural materials. Molecule branch, consisting of 6,000 beta-D glucose units joined by beta, 1 to 4 linkages, and 2,000 cellulose molecules from microfibril. Rayon and cellophane are similar to cellulose xanthate. Cellulose acetate is used to prepare tricot, double knit, wrinkle-proof and mouth-proof clothing, cigarette filters. Cellulose nitrate is used in propellant explosives. Carboxymethylcellulose is used as emulsifier and smoothing reagent. Eat chitin. Chitin is a polymer of acetylglucosamine beta, 1 to 4 glycosidic linkage forming bundles of long parallel chains like cellulose. It occurs in the cell walls of fungi and forms exoskeleton in some animals, especially arthropods. Cell walls of fungi and forms exoskeleton in some animals, especially arthropods. Pectin and hemicellulose. Pectin and hemicelluloses are structural polysaccharides. Pectins are made up of arabinose, galactose and galacturonic acid. Pectic acid is a homopolymer of the methyl ester of D-galacturonic acid. Middle lamella which binds the cells together is composed of calcium pectate. Due to this substance, water absorption capacity of wall is increased. Fruit walls contain high percentage of pectin. During ripening, the pectins break down into sugars resulting in the sweetening and loosening of fruits. Hemicelluloses are homopolymers of D-xylose linked by beta 1 to 4, xylins, arabins, galactins or hemicelluloses. These are rarely used as food, e.g. dates, phoenix. 9. Mucopolysaccharides. These are gelatinous polysaccharides formed from galactose and mannose. Slimy substances of bindi, agar, alginic acid and carrageenan obtained from seaweeds are mucopolysaccharides. Mycopolysaccharides are found in the cell walls of bacteria also. Keratin sulfate consists of acetylglucosamine, galactose and sulfuric acid, provides strength and flexibility to skin and cornea. Chondroitin sulfate consists of flyuronic acid and acetylglucosamine, present in the vitreous humor of eye, synovial fluid and cerebrospinal fluid, etc. Heparin is a polymer of alpha 1 to 4 glucosamine and glucuronic acid. It is anticoagulant present in human blood. Husk of Plantago ovata and mucilage of allobarbetans are medicinally used. Agar, alginic acid, carrageenan are obtained from marine brown algae Theophyceae. B2. Lipids. Lipids are esters of fatty acids and polyhydric alcohol. The term lipid was first used by Bloor, 1943. These are the compounds of C, H, O, but the ratio of H and O is more than 2, 1, i.e., the ratio of oxygen is less as compared to carbohydrates. These are water-insoluble organic substances which can be extracted from the cells by organic solvents such as ether, chloroform and benzene. Their general formula is CnH2nO2. Some lipids have P, N and S also. 1. Simple lipids, esters of fatty acids with alcohol. Simplest alcohol in fats is glycerol, a trihydric alcohol, e.g., fats, oils and waxes. Triglycerides are common in nature. 2. Compound lipids. These lipids contain an additional group along with fatty acids and alcohols, e.g., phospholipids, glycolipids and lipoproteins. 3. Lipids. These are isoprenoid structures, e.g., steroids, terpenes, carotenoids. 4. Fatty acids are carboxylic acid with a chain of more than 4 carbon atoms ending with COOH group. Plants can synthesize all fatty acids. Animals cannot synthesize linoleic, linolenic and arachidonic acid. These are called essential fatty acids. Their deficiency causes sterility, kidney failure and stunted growth. 5. Saturated fatty acids have no double bond. Their melting point is high. Palmitic acid, stearic acid are saturated fatty acids. Unsaturated fatty acids are commonly present in vegetable oils, cod, shark oil. Their melting points are low. Linoleic acid has 1 double bond, linoleic acid has 2, linolenic acid has 3 and arachidonic acid has 4 double bonds. Fatty acids with more than 1 double bond are called polyunsaturated fatty acid PUFA. Drying oils are unsaturated fatty acids which can be converted in hard fats on being exposed. Edible oils can be converted into hard fats through hydrogenation. 6. Waxes are esters of long-chain monohydric alcohols like cetyl, cerol or maresyl. Lanolin forms a protective, water-insoluble coating on animal fur. Beeswax secreted from abdominal glands of honeybees has palmitic acid and maresyl alcohol. Paraffin wax is a petroleum product. Cutin is formed by cross-esterification and polymerization of hydroxyl fatty acids and other fatty acids without esterification by alcohols other than glycerol. Cuticle has 50–90% cutin. Suberin is condensation product of glycerol and phenolonic acid. It makes the cell wall impermeable to water. 7. Phospholipids are triglycerides in which one fatty acid is replaced by phosphoric acid which is often linked to additional nitrogenous groups like choline, lecithin, ethanolamine, encephalin, serine or inositol. These are amphipathic i.e. have both polar and nonpolar groups. These form cell membranes along with proteins. 8. Sphingolipids have amino alcohol sphingosine. Sphingomyelins are present in myelin sheath of nerves. They have additional phosphate attached to choline, are present in nerve membrane. Cerebrosides are present in nerve membrane and have galactose. 9. Gangliosides have glucose, galactose, sialic acid and acetylglucosamine, present in gray matter, receptors of viral particles, excess causes Tay-Sachs disease. 10. Steroids contain 4 fused hydrocarbon rings called cyclopentane perhydrophenanthrene and alongside chain e.g. cholesterol, stigmasterol, campsterol, cetosterol or gasterol. Cholesterol helps in absorption of fatty acids, sex hormones, vitamin D and bile salts. Potato is rich in cholesterol. Excessive cholesterol causes atherosclerosis. 11. Prostaglandins are hormone modulators. 12. Terpenes are lipid-like carbohydrates formed of isoprene units e.g., menthol, camphor, carotenoids. 13. Major function of lipids is to act as energy stores. Plants usually store oil fats, seeds, fruits and chloroplasts are often rich in oils. Glycolipids are important components of cell membranes, chloroplast membranes. Fatty substance in the cell wall wax, cutin, suberin helps reduce transpiration and provide mechanical protection from injury and parasites. Diosgenin is a steroid obtained from the plant called Dioscorea. It is used for manufacturing anti-fertility pills. B3. Amino acids and proteins. 1. Amino acids. Amino acids are building blocks of polypeptides. Polymerization of amino acids forms polypeptides. Amino acids are linked by a peptide bond in a polypeptide. Peptide bond are synthesized during translation of messenger RNA. 2. Primary structure of a protein is the sequence of amino acids. Both peptides and polypeptides can be functional. 3. Amino acids may be functional. Various amino acids functions as neurotransmitters. Glutamate and aspartate, excitatory. Glycine, taurine, and gamma-aminobutyric acid, GABA, inhibitory. OOCCH2CH2NH3 plus GABA structure plus H3NCH2CH2 so 3 taurine. 4. Acids can act as precursors to other molecules' metabolic intermediates. Citrulline and ornithine in urea cycle can be metabolized to form glucose or acetyl-CoA. Neurotransmitters serotonin, dopamine, epinephrine, etc. Tyrosine, thyroid hormone, porphyrins. Creatine, energy storage. Histamine, mediator of immune response, nucleotide synthesis, S-adenosylmethionine. 5. Structure amino acids. All proteins are composed of 20 standard amino acids, which are specified by the genetic code. Standard amino acids are called alpha amino acids because they have a primary amino group and a carboxyl group bound to the same carbon atom, the alpha carbon. Only proline has a secondary amino group attached to the alpha carbon, but it is still commonly referred to as an alpha amino acid. The generic structure of an amino acid at pH 7 is shown below. At pH 7, the amino acid is a zwitterion, or dipolar ion. A unique side chain, or R group, characterizes each amino acid. There are 20 standard alpha amino acids, although proline is regarded as alpha amino acid. An amino acid has an alpha carbon, alpha amino group, alpha carboxyl group and a side chain, R group. These, standard, amino acids are encoded by messenger RNA. Amino acids are abbreviated by a 3-letter and 1-letter code. Abbreviations for the 20 inches standard, amino acids. Alanine A, leucine Lu L R, lysine Li K, asparaginase ASNN, methionine med M, aspartic acid As, phenylalanine Ph, cysteine CYC, proline Pro P, glycine glycerine S or S, glutamine GLN Q, threonine THRT, glutamic acid Blue E, tryptophan TRPW, histidine His H, tyrosine TRY, isoleucine EL, valine Val-V. Chemical properties of amino acids. 6. Charge. Amino acids are dipolar ions, zwitterions, at neutral pH. Zwitterion is a dipolar molecule with positive and negative charges spatially separated. Ionic states of amino acids depend on pH. Amino acids have 2 or 3 dissociable protons. pKa of the dissociable proton and the pH determine its degree of dissociation. 7. Amino acids are polymerized by condensation reactions to form a chain called a polypeptide. Polypeptide is polarized. One end has a free amino group and the other end has a free carboxyl group, referred to as the N-terminus and the C-terminus, respectively. 8. The R groups of the standard 20 amino acids are classified into 3 categories based on their polarities and charge at pH 7, the nonpolar amino acids, the polar uncharged amino acids, and the charged amino acids. 9. Among the nonpolar amino acids, glycine, shorthand GlyRG, has a hydrogen atom as its R group. Alanine Al, A phaline Pal, V leucine Lu, L isoleucine Eel, I and methionine Met, M have aliphatic chains as R groups, Met has a sulfur rather than a methylene group. Tryptophan TRP, W and phenylalanine Phe, F contain bulky indole and phenyl groups, respectively. These tend to orient inside of a protein. 10. The polar untipped amino acids include asparagine ASN, N-glutamine GLN, Q-serine SR, S-threonine THR, T-tyrosine TR, Y and cysteine CYs, C. Amide functional groups occur in GLN and ASN. Alcoholic functional groups occur in SR and THR. Tyrosine and cysteine are characterized by a phenolic group and a thiol group, respectively. 11. Hydropathy for an amino acid is an index of solubility characteristics in H2O. It combines hydrophobic and hydrophilic tendencies. It can be used to predict protein structure. 12. Among the charged amino acids, aspartate asp, D and glutamate glu, E contain carboxylate groups in their R groups. Lysine E, K arginine arg, R and histidine his, H contain a butylammonium group, a guanidino group, and an imidazole group, respectively. Both polar uncharged and charged amino acids tend to orient to the outside of proteins. 13. pK values of ionizable groups depend on the electrostatic influences of nearby groups. Inside of proteins, the pK values of ionizable R groups may shift by several pH units from their values in the free amino acids. Stereochemistry. 13. Except for glycine, the standard amino acids have asymmetric structures and rotate the plane of polarized light, thus, they are optically active. These molecules cannot be superimposed on their mirror images. Such non-superimposable pairs of molecules are called enantiomers. The asymmetric atom of an optically active molecule is called the chiral center and the molecule is said to have the property of chirality. 14. Fischer projections are used to represent the absolute configuration of substituents around a chiral center. In the Fischer convention, horizontal lines extend above the plane of the paper, while vertical lines extend below the surface of the paper. The alpha amino acid shown above is a Fischer projection of an L amino acid. The specific arrangement of substituents around the chiral carbon is related to that of L-glyceraldehyde. 15. A molecule with N chiral centers has two N different possible stereoisomers. Molecules with two or more chiral centers are better described by the RS system, in which each substituent bound to the chiral center is prioritized according to its atomic number. Hence, the exact molecular arrangement of a molecule can be unambiguously described. 16. Biochemicals almost invariably produce pure stereoisomers, in large part due to the precise arrangement of chiral groups inside of enzymes, which restricts the geometry of the reactants. 17. Non-good amino acids and polypeptides arise from post-translational modifications of the R groups of amino acid residues of the polypeptide. These modifications have critical roles in the structure and function of proteins. Many unpolymerized non-standard amino acids are synthesized by chemical modifications of one of the standard amino acids. Cells use many of these amino acids as signaling molecules, particularly in the central nervous system. Example 1. Hydroxyproline and hydroxylysine. Proline and lysine are hydroxylated enzymatically after translation. Are important in collagen structure, structures of phosphosyrine, 4-OH proline, 5-OH lysine. Example 2. Phosphoamino acids, TR, SR, THR hydroxyl groups can be phosphorylated. They are important in activation and inhibition of enzymatic or signaling activity. Among the non-standard amino acids are also the D-isomers of the standard amino acids. Many of these occur in bacterial cell walls and bacterially produced antibiotics. The tripeptide glutathione is a cellular reducing agent. 18. Titration curve of an amino acid is calculated using the Henderson-Hasselbeck equation. Isoelectric point pi is the pH at which a molecule has a net charge equals 0, average of the two appropriate pKa values. Key equation, pi equals 1/2 pKi plus pKj. 19. Amino acids TRP, TR, Phe absorb UV light between 260 to 280 nm with maxima at 280 nanometers. 20. Cysteine can form disulfide bonds. Disulfide bridges formed between cysteines are important in protein structure. Cysteine is the reduced form sulfhydryl, whereas cysteine is the oxidized form disulfide. B4 protein structure and protein folding. 1. Peptide bond. Chemically it is an amide bond between alpha amino and alpha carboxyl groups of two amino acids. Peptide bond is polar and planar. Electron resonance structure has partial 40% double bond character. Amide group is planar, usually trans. Synthesis of peptide bond involves condensation and water is produced. Energy is required for this process, which is obtained by ATP hydrolysis. 2. Peptide bond is hydrolyzable. Acid hydrolysis with 6N-hydrochloric acid heated at 110 C for 24 hours in a vacuum generates 3 amino acids, breaking all peptide bonds. Hydrolysis with 4N sodium hydroxide heated at 1000 C for 4 hours also generates 3 amino acids. Cyanogen bromide cleaves at the COOH terminal. Side of Met. Enzymatic hydrolysis of peptide bonds by proteases like trypsin cleave after COOH terminus Li or ARG. Chymotrypsin after carboxy terminus of aromatic amino acids yield peptides of various length. 3. Polypeptides are polyampholytes. Ampholyte has both acidic and basic pKa values. Isoelectric point is the pH at which the net charge is 0. For example, H3N plus allyl COO, pKa of the alpha carboxyl group equals 3.6. pKa of the alpha amino group equals 8.0. pKa of the delta amino of the lysine equals 10.6. At pH equals 1 the net charge is plus 2. At pH equals 6 the net charge is plus 1. At pH equals 14 the net charge is minus 1. Isoelectric point pi equals pKa 2 plus pKa 3, 2 equals 8 plus 1 0.6, 2 equals 9.3. 4. Nomenclature of peptides on basis of size. Dipeptide, 2 amino acids and 1 peptide bond. Tripeptide, 3 amino acids and 2 peptide bonds. Oligopeptide, several amino acids, up to 20 polypeptides, more than 20 amino acids. All proteins are polypeptides. 5. Physical forces governing protein conformation. Physical forces govern 3D structure of proteins Pauling and Corey. Bond lengths and angles should be distorted as little as possible. Structures must follow van der Waals rules for atomic radii. Peptide bond is planar and trans noncovalent bonding stabilizes structure. Conformation can change without breaking bonds. Flexibility. 6. Types of noncovalent forces important to protein conformation. Hydrophobic forces. Hydrophobic residues orient. While hydrophilic residues orient out, van der Waals potential includes electron shell repulsion, dispersion forces, and electrostatic interactions. Salt bridges, electrostatic forces and hydrogen bonds. 7. Angles of rotation of the polypeptide chain determine structure. Angles of rotation around alpha carbon are psi and phi. Psi is the angle of the alpha carbon bond to the carbonyl carbon. Phi is the angle of the alpha carbon bond to the amide nitrogen. Primary sequence and angles of rotation for each alpha carbon completely define protein conformation. Only a small number of psi and phi angles are allowed. Statistical analysis of all proteins yields groups of preferred angles. Areas of repeating psi and phi angles are secondary structures. 8. Primary structure of protein is amino acid sequence of a polypeptide. Primary structure determines three-dimensional structure. Primary structure is always represented NH2 terminus to COOH terminus. 9. Secondary structure is formed by regular local conformation of linear segments of the polypeptide chain. Secondary structure are stabilized by hydrogen bonds between amide and carbonyl groups. Several types of secondary structure. It is right-handed helix with 3.6 amino acids per turn, rise per helix 5.4 AO, rise per amino acid 1.5 AO. Here carbonyl oxygen hydrogen of one amino acid is bonded to fourth amide hydrogen NN+4. Amino acids are groups orient. Mostly proline breaks the helix. Beta pleated sheet, here polypeptide chains side by side. Polypeptide chains can be parallel or antiparallel. Carbonyl oxygen hydrogen bonded to amide hydrogen. Beta strand is a single pass of the polypeptide. Reverse turn, beta bend allows a sharp turn in polypeptide chain. Here carbonyl oxygen hydrogen bonded to third amide hydrogen NN+3. Glycine is required. 10. Fibrous proteins demonstrate secondary structure. Fibroin. Silk is fibroin which has antiparallel beta pleated sheet alpha keratin and tropomyosin. Alpha keratin occurs in woolen hair and epidermal layer. Tropomyosin is a thin filament in muscle. Both have alpha helix. Alpha helix allows elasticity. Alpha keratin converts to beta pleated sheet with heater stretching. Disulfides are important to maintenance of keratin secondary structure. Alpha keratins are beta pleated sheets in feathers and claws of birds. Collagen. It is structural protein found in skin, bones. It has triple helix, not alpha helix. It has repeating sequence GlyX Pro X or GlyX Hypro X. Glycine required for triple helix to form, contains many modified amino acids. Hydroxyproline stabilizes the structure. Vitamin C required for hydroxylation, thus in vitamin C deficiency cause scurvy. 11. Tertiary structures are overall folded conformation of the polypeptide. Physical forces affect tertiary structure. Driving force is hydrophobic force. As a result, hydrophobic residues orient to inside and hydrophilic orient out. Some extent salt bridges, electrostatic forces, van der Waals. Extent salt bridges, electrostatic forces, van der Waals radii, hydrogen bonds and disulfide bridges also play important role in maintaining tertiary structure. Myoglobin has a tertiary structure. 12. Quaternary structure are formed by aggregation of two or more subunits. These may be hetero or homo polymers. Same forces drive tertiary and quaternary structure. Hemoglobin has quaternary structure. 13. Protein folding. Folding is driven by hydrophobic forces. Folding occurs stepwise with several intermediates. Unfolded secondary structure domains molten globule native tertiary structure. A collapsed structure, molten globule, occurs very quickly. Steps between molten globule and native tertiary structure usually occur slowly. The intermediates are isolatable and there may be multiple pathways for folding. 14. Proteins can self-assemble but in vivo folding is facilitated by proteins. Chaperones are binding proteins which assist folding. Chaperones cause misfolded protein to unfold rather than aggregate. Many chaperones require ATP hydrolysis for activity. More than one chaperone may act simultaneously and sequentially in the folding of a single protein. Chaperones are specific for specific protein synthesis pathways, cytosolic versus nido. Versus endoplasmic reticulum. 15. Enzyme catalyze kinetically slow steps in folding cis-trans prolyl isomerase. Both cis and trans peptide bonds to proline naturally occur. The isomerization of peptidyl proline bonds may be slow, so this enzyme catalyzes reaction at enhanced rate. Protein disulfide isomerase catalyzes disulfide bond formation and isomerization. 16. Denaturation is unfolding. It requires some input to overcome hydrophobic forces. The heat or other denaturant, urea or guanidinium, can unfold the proteins. For reducing disulfide bridges to sulfhydryl-S, certain reductant, mericaptha-ethanol, are required. B5. Nucleic acid. 1. Nucleic acids DNA, RNA, are polymer of nucleotides. As a class, the nucleotides may be considered one of the most important metabolites of the cell. Nucleotides are found primarily as the monomeric units comprising the major nucleic acids of the cell, RNA and DNA. However, they also are required for numerous other important functions within the cell. These functions include A. Serving as energy stores for future use in phosphate transfer reactions. These reactions are predominantly carried out by ATP. B. Forming a portion of several important coenzymes, such as NAD+, NADP+, fat and coenzyme ACERCA. Serving as mediators of numerous important cellular processes such as second messengers and signal transduction events. The predominant second messenger is cyclic AMP, CAMP, a cyclic derivative of AMP formed from ATP. D. Controlling numerous enzymatic reactions through allosteric effects on enzyme activity. E. serving as activated intermediates in numerous biosynthetic reactions. These activated intermediates include S-adenosylmethionine, SAM, involved in methyl transfer reactions as well as the many sugar-coupled nucleotides involved in glycogen and glycoprotein synthesis. 2. The nucleotides found in cells are derivatives of the heterocyclic highly basic compounds purine and pyrimidine. The basicity of the nucleotides that has given them the common term, bases, as they are associated with nucleotides present in DNA and RNA. There are 5 major bases found in cells. The derivatives of purine are called adenine and guanine, and the derivatives of pyrimidine are called thymine, cytosine and uracil. The common abbreviations used for these 5 bases are A, G, T, C and U. 3. The purine and pyrimidine bases in cells are linked to carbohydrate and in this form are termed nucleosides. The nucleosides are to D-ribose or 2'-deoxy-D-ribose through AB N-glycosidic bond between the anomeric carbon of the ribose and the N9 of a purine or N1 of a pyrimidine. 4. Nucleosides are found in the cell primarily in their phosphorylated form. These are termed nucleotides. The most common site of phosphorylation of nucleotides found in cells is the hydroxyl group attached to the 5' carbon of the ribose. The carbon atoms of the ribose present in nucleotides are designated with a prime mark to distinguish them from the backbone numbering in the bases. Nucleotides can exist in the mono, D, or triphosphorylated forms. 5. Nucleotides are given distinct abbreviations to allow easy identification of their structure and state of phosphorylation. The monophosphorylated form of adenosine 5'-monophosphate, is written as AMP. The D and triphosphorylated forms are written as ADP and ATP, respectively. The use of these abbreviations assumes that the nucleotide is in the 5' phosphorylated form. The D and triphosphates of nucleotides are linked by acid anhydride bonds. Acid anhydride bonds have a high increment G0 for hydrolysis, imparting upon them a high potential to transfer the phosphates to other molecules. It is this property of the nucleotides that results in their involvement in group transfer reactions in the cell. 6. The nucleotides found in DNA are unique from those of RNA in that the ribose exists in the 2'-deoxy form and the abbreviations of the nucleotides contain AD designation. The monophosphorylated form of adenosine found in DNA DEOXYADENOSINE5, monophosphate, is written as DAMP. 7. The nucleotide uridine is never found in DNA and thymine is almost exclusively found in DNA. Thymine is found in tRNAs but not rRNAs nor mRNAs. There are several less common bases found in DNA and RNA. The primary modified base in DNA is 5-methylcytosine. A variety of modified bases appear in the tRNAs. Modified nucleotides are encountered outside of the context of DNA and RNA that serve important biological functions. 8. Adenosine, 3'-5' cyclic adenosine monophosphate CAMP, is a very powerful second messenger involved in passing signal transduction events from the cell surface to internal proteins, e.g. CAMP-dependent protein kinase pKa. CAMP is also involved in the regulation of ion channels by direct interaction with the channel proteins, e.g. in the activation of odorant receptors by odorant molecules. Formation of CAMP occurs in response to activation of receptor-coupled adenylate cyclase. These receptors can be of any type, e.g. hormone receptors or odorant receptors. Methionine, SAM, is a form of activated methionine which serves as a methyl donor in methylation reactions and is a source of propylamine in the synthesis of polyamines. 9. Guanine derivatives. Cyclic form of GMP, cGMP, also is found in cells involved as a second messenger molecule. In many cases its role is to antagonize the effects of CAMP. Formation of cGMP occurs by guanylate cyclase that is coupled to the receptor. The most important cGMP-coupled signal transduction cascade is that photoreception. 10. Synthetic nucleotide analogs. Large family of analogs is used as anti-tumor agents, for instance, because they interfere with the synthesis of DNA and thereby preferentially kill rapidly dividing cells such as tumor cells. Nucleotide analogs commonly used in chemotherapy are 6-mercaptopurine, 5-fluorouracil, 5-iodo-2'-deoxyuridine and 6-thioguanine. Each of these compounds disrupts the normal replication process by interfering with the formation of correct Watson-Crick base pairing. Nucleotide analogs also have been targeted for use as antiviral agents. Several analogs are used to interfere with the replication of HIV, such as AZT, azithromycin, and DDI, dideoxyanosine. Several purine analogs are used to treat gout. The most common is allopurinol, which resembles hypoxanthine. Allopurinol inhibits the activity of xanthine oxidase, an enzyme involved in de novo purine biosynthesis. Additionally, several nucleotide analogs are used after organ transplantation in order to suppress the immune system and reduce the likelihood of transplant rejection by the host. 11. Polynucleotides. Polynucleotides are formed by the condensation of 2 or more nucleotides. The condensation most commonly occurs between the alcohol of a 5'-phosphate of one nucleotide and the 3'-hydroxyl of a second, with the elimination of H2O, forming a phosphodiester bond. The formation of phosphodiester bonds in DNA and RNA exhibits directionality. Primary structure of DNA and RNA. The linear arrangement of the nucleotides proceeds in the 5'3" direction. The common representation of the primary structure of DNA or RNA molecules is to write the nucleotide sequences from left to right synonymous with the 5'3" direction is shown, 5' PGPA PTPC 3'12. Structure of DNA. Utilizing X-ray diffraction data, obtained from crystals of DNA, James Watson and Francis Crick proposed a model for the structure of bDNA. This DNA would exist as a helix of 2 complementary antiparallel strands, wound around each other in a rightward direction and stabilized by H bonding between bases in adjacent strands. In the Watson-Crick model, the bases are in the interior of the helix aligned at a nearly 90-degree angle relative to the axis of the helix. Purine bases form hydrogen bonds with pyrimidines, in the crucial phenomenon of base pairing. Experimental determination has shown that, in any given molecule of DNA, the concentration of adenine A is equal to thymine T and the concentration of cytidine C is equal to guanine G. This means only base pair with T, and C with G according to this pattern, known as Watson-Crick base pairing. The base pairs composed of G and C contain 3 H bonds, whereas those of A and T contain 2 H bonds. This makes GC base pairs more stable than AT base pairs. The antiparallel nature of the helix stems from the orientation of the individual strands. From any fixed position in the helix, one strand is oriented in the 5 feet 3 feet direction and the other in the 3 feet 5 direction. On its exterior surface, the double helix of DNA contains two deep grooves between the ribose phosphate chains. These two grooves are of unequal size and termed the major and minor grooves. The difference in their size is due to the asymmetry of the deoxyribose rings and the structurally distinct nature of the upper surface of a base pair relative to the bottom surface. 13. Thermal bonds of DNA. If a solution of DNA is subjected to high temperature, the H bonds between bases become unstable and the strands of the helix separate in a process of thermal denaturation. Regions of the duplex that have predominantly AT base pairs will be less thermally stable than those rich in GC base pairs. In the process of thermal denaturation, a point is reached at which 50% of the DNA molecule exists as single strands. This point is the melting temperature, Tm, and is characteristic of the base composition of that DNA molecule. The Tm depends upon several factors in addition to the base composition. These include the chemical nature of the solvent and the identities and concentrations of ions in the solution. 14. DNA, the genetic material, the physical carrier of inheritance. Friedrich Maescher in 1869 isolated DNA from fish sperm in the pus of open wounds. Since it came from nuclei, Maescher named this new chemical, nuclein. Subsequently the name was changed to nucleic acid and lastly to deoxyribonucleic acid, DNA. Robert Fuelgen, in 1914, discovered that Fuchsin dye-stained DNA. DNA was then found in the nucleus of all eukaryotic cells. During the 1920s, biochemist P. A. Levine analyzed the components of the DNA molecule. He found it contained 4 nitrogenous bases, cytosine, thymine, adenine, and guanine, deoxyribose sugar, and a phosphate group. He concluded that the basic unit, nucleotide, was composed of a base attached to a sugar and that the phosphate also attached to the sugar. He unfortunately also erroneously concluded that the proportions of bases were equal and that there was a tetranucleotide that was the repeating structure of the molecule. The nucleotide, however, remains as the fundamental unit, monomer, of the nucleic acid polymer. There are nucleotides, those with cytosine C, those with guanine G, those with adenine A, and those with thymine T. During the early 1900s, the study of genetics began in earnest. The link between Mendel's work and that of cell biologists resulted in the chromosomal theory of inheritance. Garrett proposed the link between genes and inborn errors of metabolism, and the question was formed, what is a gene? The answer came from the study of a deadly infectious disease, pneumonia. During 1920s, Frederick Griffith studied the difference between a disease-causing strain of the pneumonia-causing bacteria, Streptococcus pneumoniae, and a strain that did not cause pneumonia. The pneumonia-causing strain, the S strain, was surrounded by a capsule. The other strain, the R strain, did not have a capsule and also did not cause pneumonia. Frederick, 1928, was able to induce a non-pathogenic strain of the bacterium Streptococcus pneumoniae to become pathogenic. Griffith referred to a transforming factor that caused a non-pathogenic bacteria to become pathogenic. Griffith injected the different strains of bacteria into mice. The S strain killed the mice, the R strain did not. He further noted that if heat-killed S strain was injected into a mouse, it did not cause pneumonia. He combined heat-killed S with live R and injected the mixture into a mouse. Remember, neither alone will kill the mouse, that the mouse developed pneumonia and died. Bacteria recovered from the mouse had a capsule and killed other mice when injected into them. Hypotheses. 1. The dead S strain had been reanimated, resurrected. 2. The R had been transformed into live S by some transforming factor. Further experiments led Griffith to conclude that number 2 was correct. In 1944, Oswald Avery, Colin MacLeod, and Maudslin McCarty revisited Griffith's experiment and concluded the transforming factor was DNA. Their evidence was strong but not totally conclusive. The then current favorite for the hereditary material was protein. DNA was not considered by many scientists to be a strong candidate. The breakthrough in the quest to determine the hereditary material came from the work of Max Delbruck in Salvador. Luria in the 1940s. Bacteriophage are a type of virus that attacks bacteria. The viruses that Delbruck and Luria worked with were those attacking Escherichia coli, a bacterium found in human intestines. Bacteriophages consist of protein coats covering DNA. Bacteriophages infect a cell by injecting DNA into the host cell. This viral DNA then disappears, while taking over the bacterial machinery and beginning to make new virus instead of new bacteria. After 25 minutes the host cell bursts, releasing hundreds of new bacteriophage. Phages have DNA and protein, making them ideal to resolve the nature of the hereditary material. In 1952, Alfred D. Hershey and Martha Chase conducted a series of experiments to determine whether protein or DNA was the hereditary material. By labeling DNA and protein with different and mutually exclusive radioisotopes, they would be able to determine which chemical DNA or protein was getting into the bacteria. Such material must be the hereditary material, Griffith's transforming agent. Since DNA contains phosphorus P but no sulfur S, they tag the DNA with radioactive phosphorus 32. Conversely, protein lacks P but does have S, thus it could be tagged with radioactive sulfur 35. Chase found that the radioactive S remained outside the cell while the radioactive P was found inside the cell, indicating that DNA was the physical carrier of heredity. 15. Polymorphism in DNA. A form, having 11 base pairs instead of 10 base pairs per turn, the base pairs are not perpendicular to the axis, but are tilted. C form, like B form, but having 9 base pairs per turn. D form, like B form, but have 8 base pairs per turn. ZNA DNA with left-handed coiling is called ZDNA. 16. DA supercoiling. A supercoil is when the double helix twists around itself. Supercoils can be positive or negative, but natural DNAs exist in the negative supercoil form. DNA can be supercoiled if it is circular or if it is linear and has fixed ends. Supercoiled DNA is more compact than relaxed DNA. Negatively supercoiled DNA molecules are easier to unwind than relaxed molecules. DNA unwinding is required for replication and transcription. Topoisomerases are enzymes that catalyze changes in DNA supercoiling. A. Type 1 topoisomerases function by breaking a phosphodiester bond of one strand, passing the other strand through the break and resealing the break. They can only remove supercoil and increase linking number by 1. B. Topoisomerases function by breaking both strands and passing a double strand region through the break before resealing the break. For this process they require ATP. They decrease linking number by 2. Topoisomerases are targets of numerous chemotherapeutic drugs: adriamycin, VP16 tinaposide, BM26 etoposide. Camptothecin. 17. DNA molecule exists as single strands. This point is the melting temperature T. 17. Chromic effect. On denaturation of dsDNA into ssDNA, UV absorption at 260 nanometers increases. 18. Packaging of DNA in eukaryotes. DNA is packaged in the nucleus as a nucleoprotein complex called chromatin. Levels of chromatin packaging. A. Nucleosome core particles consist of approximately 140 BP of DNA wrapped around a protein octamer consisting of two subunits each of histones H2A, H2B, H3, and H4. Linker DNA, approximately 60 BP, is the DNA between two core particles. Histone H1 binds to the linker DNA in the core particle. B. 30 nanometers fiber, solenoid, nucleosomes are wound into a solenoid-like structure. It requires histone H1 binding to every linker DNA. C. Chromatin is the template for replication and transcription and the substrate for DNA repair and recombination. B6. Nutrition and vitamins. Nutrition is the process of acquiring energy and materials for cell metabolism, including the maintenance and repair of cells, and growth. Heterotrophic nutrition. 1. Heterotrophic organisms are heterotrophs, or organisms that feed on an organic source of carbon. The survival of heterotrophs depend either directly or indirectly on the activities of autotrophs. A few bacteria are able to use light energy to synthesize their organic requirements from other raw materials. These are called photoautotrophs. 2. The way in which heterotrophs obtain their food varies considerably. However, the way in which it is processed into a usable form within the body is very similar in most of them. It involves following processes. Digestion. This is the process which reduces large, complex food molecules into simpler soluble ones. Absorption. In this process the soluble molecules are taken away from the region of digestion into the tissues of the organisms. Assimilation. It uses the absorbed nutrients for a particular purpose. 3. Forms of heterotrophic nutrition: holozoic nutrition, saprotrophic nutrition. 4. Holozoic nutrition. The term holozoic is applied mainly to free-living animals which have a specialized digestive tract, the alimentary canal. Most animals are holozoic. The characteristic processes involved in holozoic nutrition are as follows. Ingestion is the taking in of food. Digestion is the breakdown of large organic molecules into smaller, simpler soluble molecules. Often two types of digestion occur. Mechanical digestion involves mechanical breakdown of the food, for example by teeth. Chemical digestion involves the activity of enzymes. The type of chemical process these enzyme catalysts during digestion is hydrolysis. Digestion may be extracellular or intracellular. Absorption. It is the uptake of the soluble molecules from the digestive region, across a membrane and into the body tissue proper. The food may pass directly into cells or first pass into the bloodstream to be transported to other regions of the body. Assimilation is using the absorbed molecules to provide either energy or materials to be incorporated into the body. Egestion is the elimination from the body of undigested waste food materials. Animals that feed on plants are called as herbivores, those that feed on animals are called as carnivores, and those that eat a mixed diet of animal and vegetable matter are termed omnivores. Some animals take food in the form of relatively small particles, for X earthworms and filter feeders like mussels. Some ingest food into liquid form X, aphids, butterflies and mosquitoes. 5. Saprotrophic nutrition, sapros equals rotten, trophos equals decay. Organisms which feed on dead or decaying organic matter are called saprotrophs. Other terms which mean the same thing as saprophytes and saprobiont. Many fungi and bacteria are saprotrophs, e.g. Mucor, Rhizophus, yeasts. Saprotrophs secrete enzymes onto their food where it is digested. The soluble end products of this extracellular digestion are then absorbed and assimilated by the saprotrophs. Saprotrophs feed on the dead organic remains of plants and animals and contribute to the removal of such remains by decomposing it. Many of simple substances are not used by the saprotrophs by themselves but are absorbed by plants. In this way the saprotroph provide important links in nutrient cycles by making possible the return of vital chemical elements from the dead body's organisms to living ones. 6. Protophic nutrition of Mucor and Rhizophus. Mucor and Rhizophus are common fungi known as pin molds. They are often found growing on bread, although they can also live in soil. Their hyphae penetrate the food on which they grow and secrete hydrolyzing enzyme from their tips. Enzymes carbohydrates and protease carry out the extracellular digestion of starch to glucose and protein to amino acids respectively. The thin much branched nature of the mycelium of Mucor and Rhizophus ensures that there is a large surface area for absorption. Glucose is used during respiration to provide energy for the organism's metabolic activities, whilst glucose and amino acids are used for the growth and repair. Excess glucose is converted to glycogen and fat, and excess amino acid to protein granules for storage in cytoplasm. 7. Symbiosis. The term symbiosis means literally living together. It was introduced by a German scientist Berry in 1879, who described it as living together of dissimilarly named organisms. In other words, it is an association between two or more organisms of different species. The symbiosis has been restricted by many biologists to meaning a close relationship between two or more organisms of different SPP, in which all partners benefit. Since 1970, symbiosis is more important as a topic in biology. First, we now know that the great majority of plants obtain minerals with assistance of fungi, and that much nitrogen fixation is carried out by symbiotic bacteria. Symbiosis is the living together in close association of two or more organisms of different species. Many associations involve three or more partners. Nutrition is commonly involved. 8. Three common types of symbiotic relationships are mutualism, in which both partners benefit; parasitism, in which one partner benefits and causes harm to other; commensalism, in which one partner benefits but the other receives no harm or benefit. Nutrition in humans. 9. We need food for energy, not only because it tastes good and it gives us a great feeling when we have it, but also because it gives us the much needed energy required to carry out our routine activities. Human beings require food to grow, reproduce, and maintain good health. Without food, our bodies could not stay warm, build or repair tissue, or maintain a heartbeat. Eating the right foods can help us avoid certain diseases or recover faster when illness occurs. These and other important functions are fueled by chemical substances in our food called nutrients. Nutrients are classified as carbohydrates, proteins, fats, vitamins, minerals, and water. 10. When we eat a meal, nutrients are released from food through digestion. Digestion begins in the mouth by the action of chewing and the chemical activity of saliva, a watery fluid that contains enzymes, certain proteins that help break down food. Digestion occurs as food travels through the stomach and the small intestine, where digestive enzymes and acids liquefy food and muscle contractions push it along the digestive tract. Nutrients absorbed from the inside of the small intestine into the bloodstream and carried to the sites in the body where they are needed. At these sites, several chemical reactions occur that ensure the growth and function of body tissues. The parts of foods that are not absorbed continue to move down the intestinal tract and are eliminated from the body as feces. 11. What are the nutrients that provide energy to our body and how it is measured? Carbohydrates, proteins, and fat provide the body with the energy it needs to maintain its many functions. Scientists measure this energy in kilocalories, the amount of energy needed to raise 1 kilogram of water 1 degree Celsius. In nutrition discussions, scientists use the term calorie instead of kilocalorie as the standard unit of measure in nutrition. 12. How one can classify nutrients. Is there any food which is very essential? Nutrients can be classified as essential or non-essential. Non-essential nutrients are manufactured in the body and do not need to be obtained from food. Examples include cholesterol, a fat-like substance present in all animal cells. Essential nutrients must be obtained from food sources, because the body either does not produce them or produces them in amounts too small to maintain growth and health. Essential nutrients include water, carbohydrates, proteins, fats, vitamins, and minerals. An individual needs varying amounts of each essential nutrient, depending upon such factors as gender and age. Specific health conditions, such as pregnancy, breastfeeding, illness, or drug use, make unusual demands on the body and increase its need for nutrients. Essential nutrients. 13. The most important nutrient is water. If the importance of a nutrient is judged by how long we can do without it, water ranks as the most important. A person can survive only 8 to 10 days without water, whereas it takes weeks or even months to die from a lack of food. Water circulates through our blood and lymphatic system, transporting oxygen and nutrients to cells and removing wastes through urine and sweat. Water also maintains a natural balance between dissolved salts and water inside and outside of cells. Our joints and soft tissues depend on the cushioning that water provides for them. While water has caloric value and therefore is not an energy source, without it in our diets we could not digest or absorb the foods we eat or eliminate the body's digestive was. The human body is 65% water, and it takes an average of 8 to 10 cups to replenish the water our bodies lose each day. 14. Sources of water. Many foods are also a good source of water. Fruits and vegetables, for instance, are 80 to 95% water. Meats are made up of 50% water, and grains, such as oats and rice, can have as much as 35% water. 15. Some important points. Metabolic rate BMR. It is the minimum energy requirement for maintenance of body as during rest or sleep. For a normal human adult it is 1600 kcal per day. For other animals, BMR per unit weight decreases with increase in size and vice versa, i.e., a rat will have higher BMR than a lion. Routine metabolic rate RMR. It is the energy requirement of a moderately active person. RMR is 2800 kilocalories for adult males and 2200 kilocalories for adult females. Active metabolic rate AMR. It is the energy requirement of a person doing heavy physical work. AMR is 4000, 6000 kcal depending upon the duration and intensity of work. Energy consumption per kilogram WT, 1 kilocalorie during sleep, 3 kilocalories slow walking, 4.5 kilocalories during moderate cycling, 6 kilocalories during playing, 10 kilocalories during moderate running and 20.5 kilocalories during fast running and swimming. Fuel value, amount of energy liberated during complete combustion of 1 gm of a substance in bomb calorimeter. Caloric value of fat is 9.3 kilocalories, protein 4.35 kilocalories and carbohydrate 4.1 kilocalories. Physiological fuel value, amount of energy made available during complete oxidation of 1 gm of a substance inside the body. Value per gram is fat 9 kilocalories, protein 4 kilocalories, carbohydrate 4 kilocalories. Balanced diet. It is a diet that contains all the components in optimum proportions and quantity required for maintaining the body in perfect state of health, activity and development. Various components of balanced diet are carbohydrates 60%, fats 25%, proteins 15%, vitamins, traces, minerals, traces, and roughage. 16. Carbohydrates. Carbohydrates are the human body's key source of energy, providing 4 calories of energy per gram. Carbohydrates are broken down by the body. The sugar glucose is produced. Glucose is critical to help maintain tissue protein, metabolize fat, and fuel the central nervous system. Glucose is absorbed into the bloodstream through the intestinal wall. Some of the glucose goes straight to work in our brain cells and red blood cells, while the rest makes its way to the liver and muscles, where it is stored as glycogen, animal starch, and to fat cells, where it is stored as fat. Glycogen is the body's auxiliary energy source, tapped and converted back into glucose when we need more energy. Although stored fat can also serve as a backup source of energy, it is never converted into glucose. Fructose and galactose, other sugar products resulting from the breakdown of carbohydrates, go straight to the liver, where they are converted into glucose. Starches and sugars are the major carbohydrates. Common starch foods include whole grain breads and cereals, pasta, corn, beans, peas, and potatoes. Naturally occurring sugars are found in fruits and many vegetables, milk products, and honey, maple sugar, and sugar cane. Cane starches and naturally occurring sugars are referred to as complex carbohydrates, because their molecular complexity requires our bodies to break them down into a simpler form to obtain the much-needed fuel, glucose. Bodies digest and absorb complex carbohydrates at a rate that helps maintain the healthful levels of glucose already in the blood. In contrast, simple sugars, refined from naturally occurring sugars and added to processed foods, require little digestion and are quickly absorbed by the body, triggering an unhealthy chain of events. The body's rapid absorption of simple sugars elevates the levels of glucose in the blood, which triggers the release of the hormone insulin. Insulin is in the body's rising glucose levels, but at a price. Glucose levels may fall so low within 1 to 2 hours after eating foods high in simple sugars, such as candy, that the body responds by releasing chemicals known as anti-insulin hormones. This surge in chemicals, the aftermath of eating a candy bar, can leave a person feeling irritable and nervous. Many processed foods not only contain high levels of added simple sugars, many processed foods not only contain high levels of added simple sugars, they also tend to be high in fat and lacking in the vitamins and minerals found naturally in complex carbohydrates. Nutritionists refer to such processed foods as junk foods and say that they provide only empty calories, meaning they are loaded with calories from sugars and fats but lack the essential nutrients our bodies need. In addition to starches and sugars, complex carbohydrates contain indigestible dietary fibers. Although such fibers provide no energy or building materials, they play a vital role in our health. Found only in plants, dietary fiber is classified as soluble or insoluble. Soluble fiber found in such foods as oats, barley, beans, peas, apples, strawberries, and citrus fruits, mixes with food in the stomach and prevents or reduces the absorption by the small intestine of potentially dangerous substances from food. Fiber also binds dietary cholesterol and carries it out of the body, thus preventing it from entering the bloodstream where it can accumulate in the inner walls of arteries and set the stage for high blood pressure, heart disease, and strokes. Fiber, found in vegetables, whole grain products, and bran, provides roughage that speeds the elimination of feces, which decreases the time that the body is exposed to harmful substances, possibly reducing the risk of colon cancer. Studies with fiber-rich diets, such as Africans and Asians, show that these populations have less risk of colon cancer compared to those who eat low-fiber diets, such as Americans. In the United States, colon cancer is the third most common cancer for both men and women, but experts believe that, with a proper diet, it is one of the most preventable types of cancer. Nutritionists caution that most Americans need to eat more complex carbohydrates. In the typical American diet, only 40 to 50% of total calories come from carbohydrates, a lower percentage than found in most of the world. To make matters worse, half of the carbohydrate calories consumed by the typical American come from processed foods filled with simple sugars. Experts recommend that these foods make up no more than 10% of our diet, because these foods offer no nutritional value. Foods rich in complex carbohydrates, which provide vitamins, minerals, some protein, and dietary fiber and are an abundant energy source, should make up roughly 50% of our daily calories. Dietary proteins are powerful compounds that build and repair body tissues, from hair and fingernails to muscles. In addition to the body's structure, proteins speed up chemical reactions in the body, serve as chemical messengers, fight infection, and transport oxygen from the lungs to the body's tissues. All protein provides 4 calories of energy per gram. The body uses protein for energy only if carbohydrate and fat intake is insufficient. When tapped as an energy source, protein is diverted from the many critical functions it performs for our bodies. 17. Proteins are made of smaller units called amino acids. More than 20 amino acids our bodies require, 8-9 in some older adults and young children, cannot be made by the body in sufficient quantities to maintain health. These amino acids are considered essential and must be obtained from food. When we eat food high in proteins, the digestive tract breaks this dietary protein into amino acids. Absorbed into the bloodstream and sent to the cells that need them, amino acids then recombine into the functional proteins our bodies need. The amino acids which cannot be formed by humans from other amino acids are called essential amino acids. They aid in methionine, threonine, tryptophan, valine, leucine, isoleucine, lysine and phenylalanine. 2 amino acids are slow to be formed. They are known as semi-indispensable, e.g., arginine, histidine. Animal foods, found in such food as eggs, milk, meat, fish, and poultry, are considered complete proteins because they contain all of the essential amino acids our bodies need. Plant proteins, found in vegetables, grains, and beans, lack one or more of the essential amino acids. However, plant proteins can be combined in the diet to provide all of the essential amino acids. A good example is rice lack lysine and beans lack methionine. Each of these foods lacks one or more essential amino acids, but the amino acids missing in rice are found in the beans, and vice versa. So when eaten together, these foods provide a complete source of protein.